Okay, I just want to show you a few things that I'm going to order here for this lab. We'll start with Fisher Scientific. And the first thing I want is some agarose, because in order to run electrophoresis, we need agarose. So I'll just type in agarose. Then I'll do powder because I want powder form. Agarose to channel that search. And we'll go down here. There's a few here. This one's looking good. I don't necessarily need real expensive agarose. So we'll click on here. It looks like it's about $321 each. So I'll add that to the cart. Maybe we'll go back and we'll go to the next item. So for the next item, then I want to use a different company. We will use Life Science Technologies, the same that we used the other day for getting the PCR supplies. And so for this one then, in order to stain the DNA, I need something called CyberSafe. So I'll put in cyber and then I'll put in safe. And CyberSafe then is a good DNA gel stain. So here's a gel stain here, a little safer than other highly carcinogenic things like ethidium bromide. But here's our CyberSafe. We're actually going to put this CyberSafe into our stain here. Not too bad, 400 microliters, $79, so that'll work well. So we'll basically grab that one, put that one in as well. I'll go back and show you the third item that we need. And we'll go to a different company for this third item because it's a lot less expensive. So this third item, this is actually a local Colorado company, Life Science Products, and so they give kind of supplies at a lot cheaper rate than Thermo Fisher would. So I click on here and I want something called TBE buffer. And we'll look up TBE buffer, and here's a couple different sizes, but here's a gallon jug for $59, so that ought to be good for TBE buffer. So that will work. So those are the three items that I need to order and have them come in so we can actually do this electrophoresis. Okay, the first step in electrophoresis is to prepare a gel. So when we run our gel electrophoresis, we're going to run our gels in what's called TBE buffer. Okay, so what I do is I order a 10x TBE buffer from a molecular biology company. I basically then take that 10x TBE buffer and dilute it 1 to 10. So what I do is I take one of these containers, I add 450 mLs of deionized water, and then I add 50 mLs of my 10x TBE buffer. So here's my buffer solution all ready to go. When I make up my gel, then what I want to do is I want to make up my gel in TBE buffer. So I'm going to make up 100 milliliters of my agarose gel and TBE buffer. So this is agarose. Agarose is basically a fine powder. That powder then is like gelatin. Okay, the key thing, you can actually run electrophoresis in gelatin. And there are a lot of home lab curricula where, if we did want to do this at home, you could set up an electrophoresis unit by taking batteries, taking leads, making up agarose, and then using a touch of salt and salt water, and then run your own electrophoresis. The hard part, of course, is coming up with the wells, which you'll see in a second. So what I'm going to start with then is I'm going to add, if I can get this open, I'll add 100 mLs of our TBE buffer. Now we have 100 mLs of our TBE buffer. And this container, give or take a few, doesn't matter too much. I'm then going to make a 0.8% solution of agarose. So 0.8% means I'm going to take 0.8 grams of agarose, place it in this container. So I'll put on my weigh paper, tare. Take my agarose, and we're looking for 0.8 grams of agarose. And, you know, I mean, when you run electrophoresis, we can go anywhere from 0.8 to 1%, even 1.2%, still get good results. So I'm not going to worry about being exactly right on. So there's 0.82 grams of agarose. You can see agarose is a nice fine little powder right there. So I'll take that powder, add it to my solution, spin it around a little bit. Okay, I'm then going to add an aluminum foil cap. Well, actually, I won't add aluminum foil cap because I'm putting this in the microwave. I will then basically put this in the microwave for a few minutes to melt the agarose so that I have a fully melted solution before I pour it into the molding tray. Okay, so here is my agarose and TBE buffer. Had a little bit boil out because it was hot here, so a little slightly less than 100. So I stir it around because agarose will go down to the bottom. Over here I have my molding tray. So here's my electrophoresis tray right here. I'll put that in and then tighten these up so that it's all ready to go. Okay, so these then tighten up both ends. My tray is open on this end and this end, so I have to make sure it's tight there. This is what's called a comb, so this is going to make little wells in my mold. I put my comb right there. Then I take my agarose and I pour it. So it is just a little ways up the bottom of the comb so that when it solidifies, it makes those wells. Now we leave it to solidify for about 20 minutes. 20 minutes have passed, and you can see how the agarose has solidified. So in here, it's nice and solid. You kind of see how that's like gelatin in there. Okay, the same thing has happened inside my tray. So what I'm going to do now is I'm going to gently take this comb out. So I've got to gently pry it off of those wells. And then you can see the gel here. Here's our gel. Okay, here are all of our wells. And we're then going to use the gel in gel electrophoresis. All right, so now we're ready for the next step. Okay, so here I have my DNA ladder. We have our loading die. Over here we have our 4 tubes that just came out of the fridge. These all with our PCR product right here. So AE1, B1, AE2, B2 that we did the other day. And right here I have Cyber Safe, so I'm going to add my Cyber Safe to my loading die. So it basically is SYBR Safe DNA Gel Stain, 10,000x concentration. So 1 microliter, this is 1 ml of loading dye, so 1 microliter of that with 1 ml of loading dye. So the first thing I'm going to do then is get into here. We can then see our Cyber Safe. You can see it's in a dark container because it is light activated, so it has to stay dark or it won't work. And I'm going to then get a pipette tip. Set this pipetter to 1 microliter. Alright, we'll go into our Cyber Safe. Pull out 1 microliter and put that microliter into our loading die. Okay, get rid of that, and we'll make sure that we get it all mixed up inside there so it's all in there and mixed up. Alright, so now we're all set there, so we'll take this and put it back in the box. for later use. All right. Now what I want to do is I want to basically take some parafilm. I'll cut out a little section of parafilm here. And here, let me take off that paper on the parafilm so that we can do the next step. So there's our parafilm here. And basically we're going to make up 4 samples. Right? So we're going to start with the directions. Directions say do 3 microliters of our loading dye and place it on parafilm. So we come here and we will get stop. Okay, so I want to make basically 5 different samples. Okay. And so we're going to, what I've done is I've grabbed some parafilm. I've taken the paper covering off, put it right here. Got a pipette tip. We're basically going to make 5 samples. So I'm going to add 3 microliters of loading dye to each of 5 locations on here. Alright. Now in our first one, 1, 2, 3, 4, 5, good. Our first one, then we're going to add ladder. And all of these, then we're going to add 10 microliters. So I want to change my pipetter to 10 microliters. Get a pipette tip. Get some ladder here. And I'm adding 10 microliters of the ladder to this first one. We'll mix it up and down there. Okay, number 2, then we want to add 10 microliters of our PCR product for AE1. And I will mix it up and down there, make sure it's nice and mixed. Then we will add 10 microliters of our PCR product for our B1. Mix it up and down really well here. And we'll add 10 microliters of our PCR product for AE2. And finally, last but not least, our 10 microliters of PCR product for AE2. Alright, so we are set, ready to go. Alright, so now we're ready to load the trays. So here's our electrophoresis tray, here's the gel that we made. It's in another tray there, and then we filled TBE buffer so that it just goes barely over the surface, and we have our wells right here, and we're going to run to the red electrode there. Here's our parafilm with our 5 samples. We have our ladder, AE1, B1, AE2, B2. We'll load the ladder in this side, we're going to go this direction. Okay, and we'll skip every other well just to make it easier to see. All right. So now I want 10 microliters. So I'll go in, and I'll suck up 10 microliters. And we'll add 10 microliters right there. That's our ladder. We'll add 10 microliters of AE1, skip one. And we shall put it right there. We'll get B1. Skip one and put it right there. And we'll get AE2. There. And finally then B2. And we'll put it right there, nice. Alright, now I just want to take the lid, put the lid on it, we'll attach our electrodes. And we will turn this on to 100 volts. Alright, so here we are about 10 minutes in. We can see that the loading dies can tell us where the DNA is, even though we can't visualize the DNA. And we can see the blue, so our DNA hasn't moved too far yet from our wells. It's somewhere between that blue and that yellow on that loading die. So ways to go still. Okay, our gel's done, so we're going to turn this off. We'll unhook these leads and get a view of the gel right there. Okay, and we can see that our dies let us know that it's run pretty far on here, so we're ready to go. So I'm going to take this out and then I'm going to go over and get this in the visualization device so we can get a photo of our gel. Alright, so here's our handy dandy visualization unit. Okay, so I'm going to open this up. Here's our gel. We'll place our gel face up right dead center in the middle of that guy. Close that. Here's a window if we wanted to look at it, but we won't be able to see anything. And then here I have it all set up for the camera in the back, so we're not going to quite be able to see it here, but I'll show you the picture in a second. And I'm just going to press capture. Actually, here, let's just press save. So I'm just going to press save on here, and I'm going to capture an image of that and then transfer it to the computer. Okay, so here's our gel, and we can see that the CyberSafe did a good job of visualizing that DNA under UV light, so nice fluorescent bands there. We can see our ladder to the left. We can see clearly 1, 2, 3, 4, 5, 6, 7 bands, make out an 8th one just above those values. We're looking for bands around the size of 500 base pairs, and it's looking like from the ladder that that's about right, even though we can't quite see those smaller ladder bands in there. If you look at AE1 and AE2, we can see 2 bands for AE1 and 2 bands for AE2, 1 band for bacteria 1, B1, and 1 band for B2. We can also see that AE1, those 2 bands, one of those 2 bands is archaea, the other is for eukaryotes. The band then that moves farther is our archaea, and the band that didn't quite move as far is our eukaryote. Now what we're ready to do is to go to the next step. So we verified that we have DNA in our PCR product, so we're ready to take that PCR product, send it off, and have all of those segments of DNA within those bands sequenced for later analysis.